No one was there. I thought you are also not attending class. Okay. Hello, Bhavna. Am I audible and visible? Bhavna, am I audible, visible? Okay, okay. So then I'm starting the lecture, okay? Okay. So today I'm going to discuss about the fluorescence in situ hybridization process. Okay. So in short, it is known as the fish. Okay. Okay. If you want to talk to me, you can press that hand button. Directly, you can talk to me. Okay. If you want to ask any question, you can ask. Okay. Let's start. Okay. There are few cytogenetic techniques are available. Uh, One category is conventional techniques. Another one is molecular techniques. Okay. In conventional techniques, these are a few things available. You don't have to understand about these. Now, today I'm going to discuss about only fish and gauge. Those are coming under molecular techniques, which are actually also known as the in situ hybridization. Okay. Now, what is cytogenetic? Cytogenetic means related to chromosome. So, chromosomal structure, and in order to understand the chromosomal structure or properties, if you do any study, that is known as the cytogenetic techniques. So, techniques involved for the study of structure and properties of chromosome, that is known as the cytogenetic technique. Okay. Now, today our main focus is on these two techniques: fluorescent in situ hybridization and genomic in situ hybridization. Fish and gauge. First of all, I am going to discuss about the fish, then gauge. Okay, so let's start. So now, before going to the fish, you should know about what is in situ hybridization. So, in situ hybridization is a powerful method to mainly in order to localize the nucleic acid. We do this type of we follow this type of technique. Okay, so this is actually method to localize what, nucleic acid. So in order to localize the nucleic acid, or you can consider DNA sequences in vivo. So in this case, in vivo process, you can directly localize the nucleic acid. It's not in vitro process. In vitro means in laboratory you are doing, you are using mainly test tubes, few reagents. In this way you are doing. But in vivo means directly you are using tissue or cell, and you are identifying DNA present in the cell or tissue or organelle anywhere else in the body. So, okay. So in tissue, cells, organelles, nuclei or chromosomes by using appropriate probes. So main thing is, in this process we generally use one appropriate probe in order to identify or in order to localize the nucleic acid you want to identify. Okay. That means if you want to know whether any DNA sequence is present, your interest of the DNA sequence is present in the cell or not, if you use any probe which is actually tagged with any fluorescent compound, most of the cases fluorescent compound is attached, you can localize the DNA. Now, suppose if you consider in the this is the DNA sequence present in the cell, you want to localize that this sequence is present or not. Okay, in order to do that, you have to use one probe. That probe may be single stranded DNA or single stranded RNA. So it may be the short chain. So in that case, probe is actually short chain single stranded DNA or single stranded RNA. And with this short chain DNA or RNA, generally one fluorescent compound is attached. Compound means it may be green color fluorescent compound, it may be red, multiple color is possible, available already in the laboratory. So you just tag fluorescent compound. Tagging is you have to do with the probe, okay, and you just add the probe inside the cell or during the experiment. What will happen? The probe, which has the complementary sequence inside the cell, it will bind with that only. Okay, so already you actually, in order to prepare one probe, you have to know what is the sequence of the gene you want to identify. Then only you can design one probe. Okay, artificially you are designing probe. A lot of companies are there. If you give the sequence of DNA, short chain DNA, they can prepare. It's like they can, as they can prepare primer for the PCR. In this way, they can also prepare probe, single stranded RNA or DNA, which is attached with any fluorescent compound. They can design it. Okay. So in this way, if you find any fluorescent compound, if you visualize this compound, that means actually you have localized the gene of interest or DNA of interest. Okay. So in this way, hybridization. So this compound is actually hybrid. One one DNA is present inside the cell. Another DNA you are inserting from outside. So these sources of DNA different. So this complex is known as the hybrid. Okay. Therefore, this process is known as the hybridization. So in this case, in situ hybridization. Why in situ? Because we are not changing the position of the DNA inside the cell. It is actually nucleic acids are localized in their original or proper place on chromosome. Okay, you don't have to separate nucleic acid from the chromosome. It will be situated inside the chromosome. If you consider this is the chromosome, in that case, there should be fixed position should be fixed. So you are just adding one probe here separately, which will attach with this gene, and you can identify. If you give, if you see any color, that means hybridization is done, and your gene of interest or your acid of interest is present in the chromosome. Okay, so in situ means it is localized. That means position is same. So original position is there. So nucleic acids are localized in their original or proper place on chromosome will be identified. So in this process, generally, whenever we are doing the fingerprinting or all the things, we generally separate the DNA and then you do fragmentation, and then we do SDS phase, then we do transfer to membrane, nitrocellulose membrane, and then after that only we add probe. So lot of steps are involved in case of, if you consider key DNA fingerprinting. But here, in case of southern blotting, if you consider southern blotting, DNA identification, so in that case, you are following a lot of steps. But in order to identify any nucleic acid, localization of any nucleic acid, very easily, very within few steps. Okay, if you follow very few steps only, you can identify the location of the nucleic acid through in situ hybridization technique. Okay, now what are the importance of in situ hybridization? So in this case, in situ hybridization techniques are used in order to analyze genome architecture, a repetitive DNA sequence if it is present, you can identify that. You can characterize genome. Phylogenetic analysis can be possible through the in situ hybridization. Phylogenetics analysis is relationship between the ancestors, right? We study whether relation is present or not. Now, analysis of soma clonal varieties, detection of alien chromatin, detection of chromosomal aberration, chromosome organization and interface leafly, and centromere M. Fish, centromere, that means in order to localize the centromere position, you can do fish. and chromosome painting and diagnostic cytogenetics study. In this process also you can utilize this in situ hybridization. Okay, now going to specifically fish. So till now we have discussed about the in situ hybridization. Inside the in situ hybridization, lot of techniques are there. Among them, fish is very popular. Okay, and another we are going to discuss about, that is geese. So now fish. Fish means this is a process which vividly paints chromosomes or portions of chromosomes with fluorescent molecules. Therefore, only it is known as fluorescence is given now. That means obviously some fluorescent compound is attached. So this is fluorescent in situ hybridization. In this case, what we are doing, we are actually coloring chromosome. Coloring one position of, if you want to identify the nucleic acid sequence, here localization of the DNA sequence. If you want to localize that, what you are doing, you are adding some fluorescent compound which is actually attached with the probe. So what will happen? Some hybridization will happen, and you can get, if you consider the probe is attached with the green color fluorescent compound, what will happen? In the chromosome, if you see through the microscope, what will happen? You will find green color. If you have tagged with the green color, if you used green color fluorescent compound, what will happen? This one sequence will be there, one position will be there, what will be green colored. Okay, fluorescent compound, if you added green colored fluorescent compound with the probe, so you will visualize through the microscope as the green color in this way. So this is fish means a process which vividly paints chromosome. Vividly paint chromosome means in this process I have given that only one fluorescent compound is attached. But sometimes you can see multiple sequence you can identify using multiple colorful probes. Okay, if you consider this location you want to identify, you just use separate probe with separate fluorescent compound. In this way, if you want to localize another color, suppose you are adding some this type of blue, this color, you have to prepare separate sequence for the probe, and it should be attached with separate colored fluorescent, so that at a time you can identify a lot of locations. Right here in this figure I have given 3 locations you can identify by using 3 colored fluorescent compound and 3 different probes in this way. So it is known as the vividly paint. So here we are just painting that we are giving different colors in the chromosome, adding different colors in the chromosome. Okay, so therefore this process is known as the fish. Now main purpose of doing the fish is, you can consider identifying the chromosomal location you want to analyze, or you want to identify any chromosome, you can localize that chromosome. Now in this case, most of the cases in medical use, you can consider key we can identify if there is any chromosomal abnormalities or not. So that is the main purpose. This method identifies chromosomal abnormalities, and it aids in gene mapping. So you can do the gene mapping, toxicological studies, analysis of chromosome structural aberrations, and ploidy determination. These are the applications where you can use the fish method. Okay, main purpose is we are identify chromosomal sequence. Okay, now in this process also you can do, it is used to identify the presence and location of a region of DNA or RNA within morphologically preserved chromosome preparations, fixed cells, or tissue sections. You can localize any DNA sequence, preserved sequence. Now this means you can view a segment or entire chromosome with your own eyes. Okay, so you can just identify or localize any sequence and can visualize in the chromosome. Now was often used during the M phase, metaphase, mitotic division. During the mitotic division, you can observe in the metaphase. Now, but in now used as the interphase chromosomes as well. So interphase as well as metaphase you can use. Now What are the advantages? So in this process, this is less labor intensive, as I told you, na, as compared to southern blot, it's very less, very less labor is required. Although it's very, you can consider key, you should have proper microscope in order to observe the thing, as well as few you have to use probes. Those cost of probe with fluorescent compound, those are not so to easy to construct. Okay, and price will be very high to design any probe. But it's very, you don't have to follow a lot of steps. So this is less labor intensive method for confirming the presence of a DNA segment within an entire genome than other conventional methods like southern blotting. So this is, therefore only nowadays people are not using southern blotting in order to identify any sequence, any location of the DNA or gene. We are using fluorescent compound. Now, how the preparation of biological material that has to be investigated? Acha, how means in a fish as well as in GISH method, these are the common steps involved in those methods. Like if you consider ki the first step is the preparation of biological material that has to be investigated. So you have to prepare the sample. Like if you are taking any sample from any tissue, you have to prepare that. Okay. Now probes are labeled. That means probes means here I have written in red color. Probes means short chain, single stranded DNA or RNA. You have to label it. Label it means you have to tag with the fluorescent compound. So that is the labeling. You have to label with the fluorescent compound. So preparation of that probe. Now both probes target nucleic acid are denatured. Probes and Here probes and target nucleic acid are denatured. That means whatever you are using probe in generally probe you can't add as the single stranded probe. Okay. Initially you have to prepare the probe as the double strand. Okay, single strand probe is not there. So most of the cases double stranded DNA is utilized. So in that case, if you are using double stranded DNA, which is attached with one fluorescent compound, you have to, what you have to do? First of all, you have to prepare it as the single strand one. You have to prepare single strand. Single strand, why single strand? Because in order to prepare any hybrid compound, you need single strand DNA. Okay. So you have to, first of all, we are hybridizing between what? Hybridizing between the probe and your gene of interest or chromosome of interest, right? So in that case, if you consider this is the probe, denaturation is done simultaneously. Whatever your gene of interest or DNA of interest, you have to, initially it is double stranded DNA, but you have to do the denaturation of the sample too. Okay, then only after the next step you have to mix them together so that they can make hybrid. So finally what will happen from this step, you just follow what will happen. Finally there will be hybridization of one green color which is on gene of interest, your gene of interest, and there will be bonding between probe. So these two single strands will bind together. Okay, so the single strand will bind together to make double strand. Okay, double strand. This is the hybrid one. Now, in this case, what is happening? In this case, one probe is a label is attached. Fluorescent compound is attached. Here also fluorescent compound is attached. Here also one fluorescent compound is attached, so that you can identify the hybrid because fluorescent compound is attached. So whenever binding between hybridization is done, the method hybridization is done, you have to wash the process, wash the, where you are doing the hybridization process, you have to wash it. So if you do the washing, what will happen? Only the complementary sequences will be together, bonding with together. If there is no complementary sequence with the probe, you can't find any fluorescent compound, fluorescent color. Okay, if only complementary sequence is there, then only you can visualize as the colored portion present in the chromosome. Okay, so this is the case. So single stranded probe gets hybridized to the region where it found sequences complementary to it. So this is the technique of hybridization. Then hybridization is detected and it is visualized. Visualized through the microscope. So these are the common steps involved in the in situ hybridization process. Now, if you consider this is the method of fish, in this case, fluorescent in situ hybridization, labeling with fluorescent dye, denaturation, then hybrid formation, and you can visualize it like if you are adding only one fluorescent compound and one probe, then you can get only one band, okay, in the chromosome. But if you are adding multiple color for multiple segment identification, then you can get multiple colorful bands in the chromosome. Okay, nothing else. Multiple fish. Okay, now localize here. You can find it. Okay. Simultaneously, you can identify multiple location of the DNA, multiple segments or multiple sequences. Okay, now fish uses again the detection of high concentrations of base pair. This is one example of use. That means you can identify high GC region or high AT region. Okay, region is generally, if you consider one example is given, key mouse metaphase preparation stained with DAPI. This is a non-specific DNA binding dye with high affinity for AT bonds. That means if there is any sequence segment of the sequence where lot of AT bonds are present, okay, rich in AT sequence, in that position what will bind? DAPI can bind. So you can just label it or you can use the probe as the DAPI. So it will bind at the AT rich sequence. If you visualize through the microscope that DAPI is binding with any region, that means AT rich region is present. Okay, see, this is one application. Now in the microscope you can observe in this way. See, fluorescent color is coming out of the particle here, not particle, chromosome. Okay, so multiple position here, only blue colored fluorescent compound is added, so you are finding blue color, and background of fluorescent microscope is actually black always, and background is black, but whatever color you are adding as the fluorescent compound, it will give you different. If you are adding blue colored fluorescent compound, it will be blue color, you will see blue color. Whereas if you add a green colored fluorescent compound with the probe, you can see green colored chromosome. Okay, in this way you can visualize. Now you can also identify the centromere region. So application, during the discussion of the application, I told you na centromere region also you can identify. Here I told you the centromere region also you can identify. Okay, now centromere regions stained brighter means they are rich in AT bonds. So whenever AT bond centromere, if you consider about the centromere region, na, AT bonds are more. So AT rich region is the centromere region. You can visualize that thing. Now also used in germ cell or prenatal diagnosis of conditions such as aneuploids. If there is any aneuploid is present, you can also identify through the fish method. Now aneuploidy means in this case aneuploidy means mainly uh, aneuploidy. What is that aneuploidy? I forgot. Just give me 1 minute. I'm giving you aneuploidy, mainly chromosomal abnormalities. Aneuploidy means mainly it is the chromosomal abnormalities, and it is a genetic disorder where the total number of chromosome does not equal to 46. So total in human being, if you consider Klinefelter syndrome, na? Klinefelter syndrome is there one example where generally in case of human being 46 would be the chromosomal number, But in this case, you will see genetic disorder is there. So where you can see, what you can see above 46 chromosome. See, it should be XY, but it is showing XXY. Okay, so that means chromosomal number abnormalities here. See, it should be 46, but in this case, how many numbers of chromosomes are there? 47. 46 is the normal one, right? But in this case, instead of XY, it is XXY. Okay, so this is the case. So this is called the trisomy because there should be pairing, but here three chromosomes are together. Somewhere you will see ki one chromosome is missing. So in that case also, that is also known as the aneuploidy. Okay, so another example I'm showing you. Through that also you can identify ki through the fish method you can identify ki chromosomal number abnormality is present or not. Okay, so aneuploidy, if you consider na in this way, it should be in the pair form, but it is not in pair form. Okay, monosomic is possible, double monosomic is possible. Okay, so in this case, if you consider here, this is the normal one. If you consider the four chromosomal sets are there, okay. Now in this case, trisomic, if you consider in this case, if you see now here, three chromosomes together, it should be two. Whereas in this case, doubly monosomic, here one, it should be double, here also double. So doubly monosomic. Whereas monosomic means only one present here. So one set you will find ki either it will be less than one, a less than two, or greater than two. So that is abnormality. It should be in the pair, in especially for the human being. Okay, so those you can identify whether extra chromosome is present or not. So also used in germ cell or prenatal diagnosis of conditions. So prenatal diagnosis you can do if there is any genetic disease is coming in your baby's in case of your baby. So in that case you can diagnose this before the birth. Nowadays it's allowed. If you want to terminate, if you find any genetic disorder is there, it's legal. You can terminate your pregnancy. Okay, so around 4 months, I think this determination is done. Taking the amniotic fluid, or I think something is taken, so that you can analyze the chromosomal number. Okay, so now and through fish method. Now another use is you can identify the telomeric, whether now, telomeric probes define the terminal boundaries of chromosomes. That means you know this thing, I think. Telomere. Telomere is present in human being if you consider this portion, the two end edges of the chromosome. What is present? Telomere region present. Right. So with the age, what happens? This telomere region is actually reduced. Okay. Telomere region is actually simultaneously reduced with the age. With the age, that means with the cell division, every cell division, small part of the region is actually degraded out of the chromosome. Okay. Now you can identify that region, telomere region. You can identify how much amount of telomere region is present or not, whether degraded or not. You can identify that through the fish method. So telomeric probes define the terminal boundaries of chromosomes. That means already telomeric probes are available. Okay, sequence is available, so that you can use that probe and you can apply it for the fish method. Easily you can identify full telomere portion is present or not, how much amount it is degraded. You can identify that. Now, fish method is used in research of chromosomal rearrangements and deletions related to cell aging or other genetic abnormalities. So cell aging is you can identify the telomeric region, and if you see any abnormalities or any degradation, you can identify the aging, how much it is there. You can identify how much age is happening from the telomeric portion. If you identify the telomeric portion, whether degradation is happening or not. So in this slide, I have given what is telomeric, what is happening. See, initially this is the normal case. Now slowly, small part is degraded from the telomere part. See, these are coming out of the telomere. Now telomeres, what will happen? So in this is happening due to the presence of telomerase enzyme. This type of telomere degradation, it is actually happening due to the presence of telomerase enzymes. Okay. See, the telomere itself is a long stretch of a specific short DNA sequence repeated over and over hundreds of times. So this is actually nothing. Coding in a region to any. This is actually a repeated sequence. Okay, telomeres are actually repeated sequence. In human genome, now you will find a lot of repeated sequences. So this is also telomere region is also repeated sequences are there. That means same type of sequence you will see multiple times. Here see GC, GGG, TT, GG, see GGG, TT. This sequence is present repeatedly in this portion. Okay. So this is called the repeated sequence. So at the very end of the telomere is a short not called the T loop, which keeps the chromosome ends from all sticking together. So this is uh, if you see ki side by side chromosome can stick together. So in order to control that, actually telomere is there in order to restrict the sticking together. Telomere region is there. Now every time a cell divides, some of those telomere repeats get cut off, as I told you. And so in certain cell types that divide a lot, that means if more division is happening, so telomerase, sorry, okay, I am so sorry, I told you wrongly. Telomerase is not cutting, telomerase is actually adding. So if every time his telomere part is shortened, what will happen? Very soon cell death will be there. So in order to control that, telomerase enzyme is there in our body. If telomerase is properly working, that means that cut part can be rejoined. So in certain cell types that divide a lot, in that case an enzyme called telomerase adds those repeat backs so the telomere does not get too short. Okay, so if your telomerase activity is good, that means aging will be less. Okay, because it is controlling the degradation of telomere. It is actually adding. Whatever telomeres are removed, it is actually added by the telomerase activity. Okay, up to certain age there will be telomerase activity. Okay, now if telomere is not there, that means cell death is happening. So in this figure, in the next figure, it is given if telomere is not present, so cell death is happening. See here, if you see na cell division is happening. Finally, if there is no telomere, so this region there is no telomere. Na, in the second this one, this position there is no telomere. So what is happening? So in this case, cell death is happening. Here, so this is the case. So you can identify or you can use this telomeric sequence or you can see localize the telomere sequence by Fish method. Okay. It is used in research of chromosomal rearrangements and deletions related to cell aging or other genetic abnormalities. Okay, because probe is already there, you can use that, you can identify the position of the telomeres. Now, fish and telomeres, other important thing is there. See, special telomeric probes specific to individual chromosomes have been already designed, and probe is based on the TTA-GG sequence. This is the repeated sequence in human and on all human telomeres. This repeated sequence is present. TTA-GGG. So, in order to make probe, just against this sequence, you can prepare. Okay, probe means the complementary sequence will be there in the probe. ETA GG का complementary sequence. ETA AGG. In this case, GGG, TT is given. But in human being, TTA, GG sequence is there. So if in the telomere TTA, GG sequence is there, you have to design the probe, and probe sequence will be AA, just complementary region sequence. AA, and then T, then what will be the sequence? C, and C. So in this way, you can design the probe. Already you know the telomere sequence of human. Now, this is already through the microscope, you can identify the telomere one. See, different probes, different fluorescent compounds are given for the different regions. So in this case, red color is given, blue color is given, as well as green color is given. So in this picture, you can see na telomere means at the edge, the end part of the DNA, not DNA, chromosome. It is there. So the microscope, you can visualize it. See, telomere means this part, end. This is red color probe is used. Here blue color probe is used. Here green colored probe is used. Okay. Probe means not probe. Probe is attached with the fluorescent compound. That is different colors are added. Acha. Now what is happening? Another application. Application in cytogenetics can detect submicroscopic deletions and cryptic translocations of genes associated with unexplained mental retardation and miscarriages. So, due to the few mental retardation process and due to miscarriages, what is the reason behind that? You can identify, that means by identifying the uh, identifying any gene is translocated or not. If you find that one gene is translocated from one place to another place, you can identify that through the fish method. That means if you consider in a chromosome, if you consider chromosome number 5, in chromosome number 5, in normal case there should be one, if you consider this is A gene, should be present in this position, okay, in the chromosome A, sorry, chromosome 5. But whenever you are doing the fish method, you are observing that you have used probe against this gene. Okay, you are visualizing, you are trying to localize this gene. So in normal case, you should localize these genes. You can observe in chromosome 5. But when some mental retardation or some any abnormalities is happening or miscarriage is happening, you may find that this gene is actually transferred or translocated from gene 5 to gene 21. Sorry, not gene, chromosome. From chromosome 5 to chromosome 21. So you will find the fluorescent color here in the chromosome 21. That means same probe you are using. That means same gene is localized through the fish method. Okay, it will bind the complementary sequence only. So what will happen? It should be present in the 5, but if there is any abnormalities, you will see that probe is not binding with the 5 chromosome. It is binding at the 21 chromosome. That means translocation is done here. So you can identify that. So if there is any mental retardation or miscarriage is happening and those are associated with translocation, you can confirm through this method, fish method. Okay. See, one example is given. The translocation between chromosomes 12 and chromosome 21. So this is one example is given, and it occurs at least 25% of the childhood B cell acute lymphocytic leukemia. So in case of leukemia, most of the cases you will see in the B cell, this type of translocation is happening from 12 to 21. Okay. So this is also another example, nothing else. Telomerase, see LS1, tail AML1. Yes, dual color translocation probe is designed to detect the tail and AML1 gene fusion. That means if you want to identify or localize two regions, you can do that. I told you you have to use two different probe which are attached with two different fluorescent compounds. Okay. So you will find key different colored labeled different labeled site will be there. Okay. So in this case, one example is given key probe is used against telomere, tail, and AML1 gene. So this fusion is undetectable by standard cytogenetics. Through the cytogenetics, you can't normal standard cytogenetics you can't identify, but you can identify it through the visualize is through the fish method. Acha. Now, fish can be used in the study of transgenic animals. Transgenic animals, mainly you can consider the recombinant animal. If you are inserting any gene from human, you are taking gene from the human and inserting in any mouse. That means if expression is happening, so that is known as the transgenic mouse. So recombinant mouse, if you consider, you can identify whether human gene is inserted in the mouse or not. You can identify that. The selective markers show if the human DNA was inserted successfully and in pinpoint where the human DNA is. So that is the importance of hybridization fish method. You can visualize exactly which position this gene is present. So if you are inserting one gene which is coming from human being, in situ process is so beautiful. You can very easily identify that gene inside the transgenic mouse. You pinpoint, you can just point because you are using probe against that human gene. Already you have that sequence of the human gene and you can design probe. You can add one fluorescent compound with the probe and you just do the hybridization process. Okay, you can visualize if you are getting any color, that means fluorescent color. If it is visible, that means it is the so transgenic mouse. In the transgenic mouse very successfully DNA is present, human DNA is actually present inserted in the transgenic mouse. Okay, in this way, see here a lot of visualization is given. Here red colored my fluorescent compound is used. In this case, all the chromosomes are not fluorescent color is giving. They are not giving all the chromosomes are not giving any fluorescent. See this color is this is giving, this is giving, this is giving, and this part is giving. So these are showing that because you are using specific gene probe against these specific region. Okay, in this way we can visualize. Now going to talk about the in this method you can use direct method, you can use or indirect method you can use. You can detect okay fluorescent compound during the discussion of ELISA. I don't know whether you have joined my class at that time or not. During the discussion of ELISA method, I discussed various methods of ELISA. Among them, direct and indirect method I have discussed. Direct method means generally we use most of in if you consider about the ELISA technique, if you are using any anti and you are identifying any molecule, okay, you just directly add probe and that probe is actually attached with the fluorescent compound. Whatever I have discussed till now, this is actually I have discussed about the direct method. That means directly fluorescent compound or radioactive compound is attached with the probe and we are observing the hybridized molecule. Whereas in case of indirect method, what is happening? In that case, you are using one, this is the gene of interest. Now you have to use one probe. Suppose this is the probe. Okay. With probe, most of the cases what is attached in this case and one attachment is there, that is called the biotin. Okay, if you consider key your probe is attached with the biotin or deoxygenine di digoxygenine, okay, or fluorescein. See, in this case, mainly biotin is most of the cases used. Either biotin or digoxygenine is used. So, but biotin is not giving any color because this is not fluorescent color. Okay. So, against biotin, you have to use something. Okay, in that case, most of the cases against biotin, generally what is used? What is used? One antibody like structure actually, that is mainly avidin is used. Avidin, yes. What is used? Just a minute. Biotin. In this case, generally avidin, yes. So against that, you have to use avidin, and this avidin is actually attached with the any fluorescent compound. So directly fluorescent compound is not attached with the biotin, okay, or in the probe. Some secondary thing is there. So during the discussion of ELISA, I told you one antigen is there. You want to identify any antigen. So in direct method, what you will use? You just use one antibody against this green color, and this antibody is actually attached with the fluorescent compound. This is called the direct method of ELISA. So not fluorescent. In ELISA, we label this antibody with the enzyme. Okay. Now in indirect method, what will happen? In indirect method, what is happening? This is the antigen. You want to antigen. So this is the antigen. Now against that, you have your primary antibody. This is called primary antibody. Now primary antibody is there. Uh, Against primary antibody, you have to use one secondary antibody. So in this case, FC region of the primary antibody is actually attached with the FAB region of secondary antibody. Now this is secondary antibody, blue color is secondary antibody and red color is primary antibody. Now with secondary antibody, you have to add the fluorescent compound. Okay. So it was the indirect fluorescent enzyme is attached for the ELISA technique. So in indirect technique, generally we are directly not adding any fluorescent compound in the with the probe. We are only adding. You have to add some primary thing. Against the primary, you have to add secondary thing, and with secondary, there should be some binding of fluorescent compound, or in case of ELISA, it is enzyme. Okay. So this is happening. So in direct means directly you are adding the fluorescent compound with the probe. In indirect, you are using something in between the in between the probe and fluorescent compound. Okay, in this way it is happening. Now, so in this case, in case of direct method is more popular because it is giving you the better result. So detection will be better in the indirect method. So there are two methods for multicolor fish. So the indirect method uses biotin and digoxygenin and dinitrophenol. Another compound is given here, DNP, as the reporter molecule. So in this case, it is known as the reporter molecule. Okay, whereas if you consider about the ELISA method, in that case we are talking about the primary antibody. Okay, in this case it is reporter molecule. Biotin, digoxygenin, those are actually reporter molecules. Now they are detected by fluorochrome conjugated avidin or antibodies. Okay, so antibodies will be against degoxygenin. So that means degoxygenin is like, I think, protein antigen. So against that, there will be antibody. So anti-degoxygenin. So you have to add fluorescent compound with avidin or anti-degoxygenin. Now fluorochrome labeled nucleotides are used for probe labeling in the direct method. Whereas in the direct method, generally we are using fluorochrome labeled nucleotides as the probe. Now, direct coupling of reporter molecules like fluorochromes to probes eliminates the need for immunocytochemical detection. Thus, the direct method has two advantages over indirect method: better resolution and speed. Speed will be better, but I think more indirect method is most popular, generally used. So, directly labeled means fluorochrome is directly attached to the probe nucleotides, whereas indirectly labeled means via incorporation of a heptane, that is biotin or digoxygenin. Okay. So this is actually biotin. If you consider, this is the ash color, this is biotin which is attached with the probe, and biotin is attached with the avidin. Okay, avidin or digoxygenin. and with digoxygeny, sorry, I'm wrong. Ash color, this ash color is, or I'm giving you the blue color. This blue color is actually biotin, which is biotin or deoxygenin, which is attached with the probe. So against that, you have to add something which can bind with the biotin or digoxygenin. So that is actually known as the avidin. If you are using biotin, so against that you have to use avidin. If you are using the digoxygenin, so in that case you have to use anti-digoxygenin. Okay, so antibody against digoxygenin. And with anti-digoxygenin or avidin, what is attached? One fluorescent compound is attached. So this is called the indirect method, and this is direct method. Directly fluorescent compound is attached with the probe. So this is all about the fish. Now what are the steps involved in this process? What are the steps involved? Let's talk about those things. So in this case, what is happening? If you see, just a minute. So up to this, is there any confusion till now? Till this PPT, is there any confusion, Betu? Tell me. Whether is it clear? Are you understanding what I am talking about? Bhavna? Hmm? Let me know whether are you understanding the topic or not. Beta? Okay. Uh, so what are the steps followed here? So let's see. Okay. So first of all, this is so you have probe and your gene of interest. You need these two things. So now to prepare those things, you have to prepare the probe. Na? You have to prepare the probe. You have to prepare the gene of interest. So in that case, you have one gene against, and that means you have initially you have double stranded long gene. Okay, and in order to make it as the probe, you have to degrade it first. So because probe is generally, I told you, ki small fragment should be there, small stretch, less number of nucleotide sequence will be there, should be there. Probe, that will be better attachment with the gene of interest will be better if you use small fragment of DNA or RNA. So now to get that, you have to do the fragmentation of gene. Okay, so in order to do that, you have to do the fragmentation of gene, and in order to do that, you have to add DNase enzyme. DNase enzyme will randomly cut few. It will remove TT region, TT portions, and now DUTP for biotin, GUTP. Now you just add the biotin compound. Biotin for the, if you consider ki direct, you are following the direct method. No, indirect method only. You are using the biotin, na? So you add biotin so that biotin can attach with the UTP. Uracil region will be there, so that if you are using any RNA molecule, uracil labeled biotin, so there will be addition of the biotin molecule in the probe. Okay. Now side by side we have to do key. You have to first fix the cell. If you want to localize one chromosome which is present inside the cell, first of all you have to fix the cell. Okay, fix the cell or fix the tissue from where you are identifying the DNA position. So you have to fix the cell, okay, on the slide. In this case, in microscopic studies, so you have to fix the cells on the slide, glass slides. Okay. Now, simultaneously, you have to do denaturation of the gene of interest, chromosome of interest, as well as you have to denature the probe. Clear? Acha, in this case, for the I told you wrongly here. Just a minute. I want to repeat these sequences again. Just a minute. So, first of all, you have taken one gene. You want to make it as a probe. Okay. So, in order to do that, you want to add biotin in the sequence. Okay. And you have to add the biotin, no? Because in order to use as the probe, you have to add either biotin or directly you can label it. Now, here it is the indirect method. So, biotin should be added, and biotin is actually added with the UTP region. Okay. Now, in order to do that, what is happening? This double stranded small fragment is from that some DNAs is given so that few T regions can be removed. So, specific for the region removal, it is given. You can consider, or few other may be possible. If few other sequences also deleted. Single stranded, mostly single stranded degradation is happening here. So, and you just adding what? You are just adding biotin labeled DUTP plus you are adding CTP, ATP and GTP. So, total four bases you have added along with, and among the four bases, only one is labeled with the biotin. Okay. So, what will happen? Biotin is only labeled with the U. Okay. So this is, I told ki complete fragmentation is done first. No, complete fragmentation is not done. If you are taking already you are taking small fragment, you have designed already. You have designed in the laboratory. A lot of companies are there. If you give the sequence, they can design this double stranded short chain DNA or RNA. Okay. So now what you have to do, you have to denature both the gene of interest or chromosome of interest, or you have to denature probe. Okay. Denature in order to make it single stranded. Then only they can bind together to make the hybrid. So this single strand, this is single strand. Now they are binding together to make the hybrid. So this is our actual interest. If whatever probe you are using, if complementary sequence is present in this gene, that means it will bind with the your chromosome of interest or DNA of interest, है ना? So this is the hybridized DNA, and this hybridized DNA, what is attached with? So biotin is attached with this hybridized DNA. This is, these two biotin molecules are there. Now what you have to do, you have to visualize it. Na? Biotin cannot give you any color. So you have to visualize ki whether hybridization is happening or not. In order to do that, you have to add antibodies or avidin plus one fluorescent compound. So this is antibody or avidin plus one fluorescent compound. So they should attach together, and what will happen? It will make this complex. So in the hybrid, what you are visualizing through the microscope, that is actually DNA and probe hybrid plus biotin plus avidin and plus fluorescent compounds. So, so many things. So you can consider 1, 2, 3, 4, 5. 5 things are actually attached. Okay. So what is present? Actually, I'm writing here one DNA. So what you want to identify the sequence. So if you consider this is the sequence you want to identify in the chromosome. So against that sequence, already you have added what? You have added one probe. So hybridization will happen, and with probe what is present? One biotin compound present, one A two biotin present, and against biotin you have to add avidin or any antigen, and with avidin what is present? Fluorescent compound. Okay, so I am writing here ki red color fluorescent compound is attached. So this is actually, this is the complex what we are observing through the, or visualizing through what? We are visualizing through the microscope. Okay, na? After the washing, what will happen? Only if probe is binding with the chromosome, you can visualize that position. If it is not binding, probe is not binding, you can't find any color. It is because complementary sequence is not there, therefore it is washed out of the slide. Okay, na? So this is the case. Acha. Simultaneously, let's see the use again. The many genes are similar in most plants, and the ordering of these genes is highly conserved across wide taxonomic grouping. In wheat and rice, linear order of markers in linkage group is same. So you can visualize the similarity is there or not in between two species. Okay, see wheat and rice, there should be some similarities. Okay, na same group में आ रहा है taxonomically. So many genes are similar in most plants. So similarity you can identify, dissimilarity you can identify through fish method. Now, if you consider here detection of chromosomal aberrations. So multiple, if you add the multicolor probe, you can visualize a lot of segments in the chromosome. Fish can provide a rapid and accurate identification of most common trisomics and sex chromosomes abnormalities, structure and abnormalities. I told you already about the aneuploidy, so you can identify that. In many polyploid species, there are intergenomic translocations shown by fish. So translocation also you can visualize almost aberration observed during meiosis. So meiosis also you can visualize. Now my advantage of multicolor fish demonstration of a structural aberration which cannot be detected by the conventional staining method. So what through the conventional staining method you can visualize multiple regions only through this in situ hybridization phase method. Now, simultaneous visualization, in this case, another example is given. Simultaneous visualization of total genomic and highly repeated DNA as probe is also useful for investigation of chromosome organization in interphase nuclei, orientation of telomeres and centromeres. Already I have discussed this thing. Same thing is written here, nothing else. And in this figure, you can see telomere region you can identify and centromere too. Mostly, this is centromere. See, this is actually centromere. You can visualize properly. You give separate color for the centromere. You can give separate color for telomere. 80 is region telomere. 80 is there also. 80 is region telomere is also 80 region. Same strain also you can use, but probe will be different. Okay. In this case here, centromeres are there. Visualize. You can visualize this. Telomere part is there plus centromere part. Both you can visualize. Okay. Now plant telomeric sequences have been cloned from. So this picture is from the Arabidopsis thaliana. Okay, centromeres and telomeres can identify. Now, centromere specific multicolor fish is a new technique that allows the simultaneous characterization of all human centromeres by using labeled centromeric satellite DNA probe. So that means you can label all the chromosomes and all the centromeres. The sequence will be different for different chromosome. To some extent, difference will be there, and you can use separate probe. So much of 23 sequence will be there. Okay. Total chromosome 23 na pairs. So already sequence probe sequence is there against that, against the separate centromere. So you can use that and identify all the sequences together. Acha, now chromosome painting. Same thing, chromosome painting. You can make colorful chromosomes by staining, by adding different fluorogenic compound. See, by mixing combinations of 5 fluors and using spatial imaging software can distinguish all 23 chromosomes. So you have already probe with different colored fluorescent compound. Okay. Now whole chromosome or region specific paints can be produced by interspersed repetitive elements, and chromosome specific probes are now commercially already available hai for all chromosome that can be used in prenatal and diagnostic cytogenetics. So if you want to check any abnormalities is there in the baby abnormalities in the condition of prenatal condition, you can diagnose that. See, few repetitive sequence you can identify. A lot of repetitive sequences are there, and in humans especially you can consider. and but repetitive sequence will be different for different person. See, following are various classes of repetitive DNA sequence. Where repetitive sequences are there, mainly you can observe it in the rRNA genes, in the tandem repeats, telomeric sequence. I told you already, in the telomeric sequence, repetitive sequences are there. Centromeric sequence, they are also repetitive sequence there, and in the macro satellite sequence. So microsatellite is small fragment repetitive sequence, small less than 10 base pair or less than 6 base pair repetition will be there. That is the microsatellite. Tandem you can consider is slightly, I think, higher than that, above 10 base pair repetition. Okay, so this is all about fish method. Okay, so now I'm going to talk about Gish method. So, so fish method, what we understood from this discussion, that mainly in fish method we are visualizing chromosomal sequence. We are comparing chromosomes. Okay. So if there is any problem in the chromosome, you can identify that by fluorescent labeling using the probe which is actually attached with fluorescent compound. Right. So we are mainly focusing on the chromosome. Okay, remember it. For the fish, we are actually focusing on we are staining, or you can consider fluorescent staining, we are doing in the chromosome. So visualizing the chromosome, these are all the chromosomal structure. Chromosomes are visualized. Okay, this is also chromosome, na? Chromosomal picture. The fluorescent in situ hybridization is actually we are focusing on chromosome. Now going to next topic, that is we are talking about the, now I'm going to talk about GAZE method. GAZE method is actually genomic in situ hybridization. Okay, so is there any doubt from the fish method? Tell me, then I can continue with the GAZE method. Beta, you understood now what actually I'm trying to tell you? Bhavna, fish method application you can understand? You understood the fish method, huh? Shraddha, you left the class, I think, for some time. Bhavna and Shraddha, tell me, is there any doubt? You can directly talk to me. If you want to talk to me now, you just press the hand button and tell me if you want to ask any question from fish, whether you understood or not, tell me. Shraddha, beta. Okay, no problem. I'm continuing, okay? Should I continue or not? I'm continuing, okay? Gish method means genomic in situ hybridization. Genomic. From the name, you can understand what actually going to happen. So GACE means genomic in situ hybridization. In fish, we are focusing on the chromosome, right? But in GACE, we are actually focusing on the genome. Genome means genome means total DNA sequences present, total genes. To all genes together is known as the genome. So we are focusing on a whole gene, complete genomic sequence. So genomic in situ hybridization. In situ hybridization is same. That means you want to localize one sequence, identify one sequence, and you already have probe, okay, against that, and with probe, if you following a direct method, Then with probe, one fluorescent compound is attached, and you are just making the hybrids, and you are visualizing that. That is the basic concept for the in situ hybridization that is happening here also. But in fish, we are focusing on the chromosome. In this case, we are focusing on genome. Okay. Now see, GISH uses genomic probes. So in this case, genomic probes used that are prepared by cleavage. So actually, if you consider these are the different, if you consider this is the genome sequence, now you have to make fragments of the genome sequence, and then you can use the probe. So you have to make different fragments from the genome first, then only you can do the hybridization. So GISH uses genomic probes that are prepared by cleavage and fragmentation of genomic DNA, usually from a parent hybrid, so allowing the in situ detection of the genome and is homologous to the used probe, allowing the in situ detection of genome. So you can detect the genome in situ, and that is homologous to the used probe. Whatever probe you are using, it should be homologous to the genome. Then only you can identify that genome. हाँ चली गई. Okay, so okay, genomic in situ hybridization is modification of fluorescent in situ hybridization. That means GIS is related to fish, but just some modifications are done in the GIS method. And GIS for plants was developed by Bennett and Heslop Harrison. Okay. Now, this is a cytogenic technique, obviously cytogenetic technique that allows the detection and localization of specific nucleic acid sequences. Here also we are localizing specific sequences on morphologically preserved chromosomes. Okay, using genomic DNA. So in this case, we are localizing actually, you can localize morphologically preserved. Some preserved chromosome will be there, chromosomes using genomic DNA of donor species as the probe. Now, GIS is technique that allows distinguishing the genome in a cell. So if you want to compare genome between species A and species B, you can compare it by GIS method, because whole genome, you have the whole, you have to compare between the two whole genome sequence, and that is done through the GISH method. So species to species, there will be genomic sequence difference will be there, you can identify that. Okay, so mainly GISH method is used for the evolutionary studies. If that means, if you consider you have taken species A genome and species B genome, okay, you are analyzing. If you find there is 100% similarity in the genomic sequence of A and B, that means you can consider A and B actually coming from the same species. Okay. Now, if there is some similarities, few similarities are there, that means you can say it is related. They are related species, or they are coming from the same ancestor or not. You can analyze the phylogenetic relationship between species to species, okay, through GIS method, because we are comparing the genome. Now, an unlabeled DNA of parental species is used as the competitor DNA. So, in order to do that, one genome we are labeling, another one we are actually not labeling. If you consider we are checking one hybrid, okay, hybrid genome. So, if you consider this is hybrid genome. So, this hybrid genome is actually coming from where? Two separate genome. So this is blue color and another is coming from the green color. Now you have to use blue color as the competitor DNA, which is actually not labeled, and you can use this green color as the probe, which is actually labeled. So, so what will happen? You can get one hybridized compound which is actually labeled with this probe, and you can compare ki whether same type of genome is present or not. Same type of genome is present here in the hybrid or not. Okay, sequence similarity is there or not. You can identify that. Okay, now what are the uses? So it is possible to differentiate the genomes in a hybrid. See, already I told you you can differentiate the genomes in a hybrid to the study of hybrid lineages, detecting the amount of introgressed chromatin during the production of new lineages. So lineage means new organism, but from the same there is some relation is there. Okay. Now evolution of polyploids, whether mainly this is utilized for the evolutionary studies. Evolution of polyploids. Polyploid is how it is developed. You can identify that. You can check that. Now, fish has direct applications on the, oh, sorry, not fish, fish has direct applications on the understanding of genome evolution. So, mainly we are observing genome structure. So, you can find out the evolution of partial alloploids and recombinant inbred lines. So, if there is any recombinant happening, hybridization is happening, you can identify that. A GIST can be applied to the analysis of mutatic behavior in hybrids. In the hybrids, already same thing is given, nothing else. So in hybrids and polyploids, providing information concerning the relationship between species. So phylogenetic, you can consider a phylogenetic study you are doing. GIST is used for purpose of distinguishing chromosomes from different genitors. Genitors means different genitor means same origin, or from different genomes in interspecific or integratic hybrids or alloploids. So alloploids means an individual that is hybrid of two different species. Okay. So what I told you about this thing, this is actually blue and green combination is there. Genomic combination is there. This is actually you can consider a hybrid hai. Now an individual that is hybrid of two different species and that possesses more than two sets of chromosomes. So here, not only hybrid, in that case, two sets of chromosomes, more than two sets of chromosomes are present. In that case, it is known as the alloploids. So you can identify that alloploidy. So what is alloploidy and autoploidy? You should know that. Autoploidy means polyploidy. Actually, you can categorize into two types. One is autoploidy and another one is alloploidy. Okay, autoploidy means you are taking more than one chromosome, more than pair chromosome present, but those are coming from the same species. That is the autoploidy. But if you consider with the polyploidy, alloploidy, in that case, the combination of genetically distinct but similar chromosome sets. So that if you consider, I don't know whether it is visible or not. I'm giving you the separate slides so that you can visualize what is the difference between the alloploid and autoploid. If you consider this one, I'm giving you separate figure here. See, in this case, initially, if you consider species A, this is the 2n chromosome number, 2n is equal to 4, and in this case it is species B, 2n is equal to 6. Okay, n means pairing, how much amount of pairing number paired chromosome there. So in that case, if in the normal case, what will happen from species B, if normal is happening, normal meiosis is happening, what will happen from 6 phase 6 chromosome, you will in during meiosis process, 2n is converted to n. That is happening in meiosis process. Meiosis means always 2n chromosome number is converted to n chromosomes. Reduction of the chromosome number is happening during the gamete formation. So this is actually gamete. So in this case, initially it was 6 number, and during the gamete formation, what is formed? n is equal to n. Okay. Now, if you consider the species A, if there is any abnormality during the meiosis process, this reduction is not happening. That means from one cell to how when gamete formation is happening, due to abnormalities, what is happening? Same number of chromosomes are present. 2n from the 2n, 2n is there. Okay, so it is not converted to n number. So what will happen? Some more after the fertilization, what will happen? This 4 plus 3, there will be total, this type of combination will be there, hybrid in the hybrid. But in the normal case, it should be 2n number. It should be twin number. It should be 4. So it should be 2, total 2 number, and it should be 3. So that 2 plus 3, it should be 5 in normal case. But in this case, it is actually in case of alloploidy, what is happening? Some abnormalities is there. Abnormality means number of chromosome increased here. It should be 5, but it is giving you the 7 chromosome in the gamete. Okay, so here see normal, not gamete, in the fertilized thing. So unreduced gamete 7 hai. Now combination of these things, so 7. So you will get, finally you will get, if hybridization is happening in between these two, what will happen? There will be combination of these two things. So 7 plus 3, that is total 10. So in this case, it is giving you the total 10. So viable fertile hybrid, this is actually, it is converted to total 10. Okay, some abnormalities. So it should be 10, no? Finally, it should be 10 species. And what is gamete hybridization? And then, acha. Okay. So in this case, what is happening now? First of all, we are making one hybrid. We are making hybrid first. So in this case, it is 7. It should be 5, but in the hybrid, you are getting the 7 chromosome. And then with the 7 chromosome, you are doing the fertilization with normal gamete, that is 3. So finally, you are getting 10. Okay. So normal case also, you can get total 2n is equal to 10, because if you do the hybrid meiosis of species A and meiosis of species B, what will happen? Finally, you will get total 2n. After the fertilization, what will happen? In the zygote, you will find 2n equals to 10. Okay, that is normal case also happening. But if you do through the hybridization, this will be the process. Middle step is there. Okay, so this type of combination is known as the alloploidy. So here see the combination of the genetically distinct but similar chromosome sets. So here we are combining two genetically distinct but similar type of chromosome. Hybridization is done. Now autoploidy. Autoploids are derived from within a single species. So in this case, there will be species A with species A. So this is species A and A. There will be the case, not species A and B. Separate species there will be not happening. It is not happening in the autoploidy. So again, I am going to show you the actual figure. What is autoploidy? Individual has greater than 2 chromosome sets, all derived from a single species. In that case, so it is derived from single species, but greater than chromosome 2. Okay. So now you, what, why I have discussed that? Because it in case of GISH method, uses of GISH method within this topic, uh, alloploidy is coming. So GIS is used for the purpose of distinguishing chromosomes from different genitors or from different genomes in interspecific or intergeneric hybrids or alloploids. So if there is any hybrid or alloploid is happening, you can identify that. Okay, through the GISH method. So now what are the steps involved in this process? Steps involved in GISH you can consider ki initially it's although similar to fish, but few new things are happening in this process. Like you have to prepare, first of all you have to prepare the probe. So probe DNA, then isolation and sharing of the DNA. Okay, probe DNA preparation. Now isolation and sizing the competitor DNA. So in this case, what I told you initially, I told you during the discussion of the definition, I told you ki in this process what is used, not only the probe is used, simultaneously you have to use one competitor DNA, and that is not probe. That means fluorescent is not labeled with that. Okay, here it is written an unlabeled DNA of parental species is used as competitor DNA. So this is the new thing happening in the GISH method. In fish method, we are not using it. Unlabeled DNA parental species, we are not generally used. Okay, but in GISH method, we are using. Now, nick translation labeling of the probe DNA. Okay, so nick translation already I discussed that you are removing few strands from the few nucleotides by DNA treatment. Okay, and then you can label it with the biotin if you are using the secondary, or if you are using the indirect method. You can prepare the probe, or you can label the probe by nick translation labeling of the probe DNA. Now purification of labeled DNA probe. Now you have to purify the probe. Then chromosome preparation. Now next step is the chromosome. Simultaneously you have to prepare a probe, you have to prepare chromosome. So chromosome preparation, in situ hybridization. Now already probe is there. Chromosome you have to degrade into few fragments. Whole genome you are analyzing. So you have to degrade the genome, and now this DNA probe will attach with the gene of interest, and that means hybridization is done. Then finally you have to do the detection of hybridization and microphotography you can take. Okay. Now DNA from a test organism that is denatured and then used in vitro hybridization experiments in which it competes with DNA from a reference organism. So in this case, as I told you, another DNA you have to use. It's like probe, but not probe because it is not labeled with any fluorescent compound. So that is actually source is actually a reference organism, and those are used to determine the relationship of the test organism to the reference organism. We are all checking relationship between the test organism and the reference organism. Okay, that is it. Okay. So in this case, major difficulty is in GACE method is that this difficulty lies in the adjusting the optimal concentration of blocking DNA in such a way to enable genomic distinction without non-specific hybridization. So in order to avoid the non-specific hybridization, what we are doing? Blocking the DNA by using separate DNA sequence. Okay, and engaged the blocking DNA should preferably consist of non-target genome. So whatever genome you want to identify or localize, you you should not use reference, or you should not use the blocking DNA against that complementary sequence. Okay, so in GACE, the blocking DNA should preferably consist of non-target genome. Thus, the blocking DNA will compete with the genomic probes. So there will be, we are mixing probe plus blocking DNA. So there will be competition between probe and blocking DNA. Okay, so now if sequence against the probe is there, you will find fluorescent color, enabling a specific hybridization. Due to this competition, there will be specific hybridization. There will be no non-specific hybridization. So here and there, their binding is not happening. Here, self binding is not happening. Cell bond formation is not happening. So non-specific binding is not happening. In order to do that, we are using blocking DNA in this process. Okay, now so in this process, DNA of a genitor involved in the formation of hybrid is used to the same time as an unlabeled DNA. So this thing, DNA, what you want to make the hybrid, so you have to use another unlabeled DNA from another genitor at a higher concentration. Generally, this blocking DNA is utilized at the higher concentration, which serves as a blocking DNA, and hybridizing with the sequences is common with both genomes. So it will hybridize with the sequences which is actually having the common genomic sequence. Now, total genomic DNA must be broken into smaller fragments. So, if you see the steps involved in this process, now you'll see first of all you have to do the fragmentation of the genome, which can be done by using restriction endonucleases or by autoclaving or sonication. Okay, and this case blocking of the DNA is necessary. Now, due to the high degree of homology shared between the species that originating the hybrid, which may enable the probe to label the genome of both genitors distinctly. So, if you want to identify the separate genitor, you are checking whether similarity is there. In order to avoid the non-specific hybridization, you have to use blocking DNA. Okay, one example is given, nothing else. Now, by adjusting the blocking DNA concentration for each species, okay, in accordance with the degree of genetic proximity, it is possible to assure that probe stains only one of the genomes. So, probe whatever you are using, only it is staining one genome, not the both of the species. Another species specifically you can consider key, another blocking DNA is actually binding with that. So this is the case. You just try to study it. I'm coming back within 1-2 minutes. You just read this slide. Okay. Bhavna, you just go through it. This PPT, I'm coming within 1 minute just. Okay. So process it is written in this slide. Okay. Now, so in this case, what is happening? First of all, if you are using direct method, you just add the probe which is labeled with the fluorescent compound. Now, if you are using indirect method, you have to follow that process. Biotin addition or other compound should be added. Okay, so indirect method or direct method preparation. Now what you have to do? Probe denaturation. So next step is the denaturation. Always if you want to make the hybrid molecule, you have to do the denaturation process. So probe denaturation is done. Okay. Now I'm going to the next. That is fragmentation of blocking DNA. So what I told you, in this process you have to use another blocking DNA. Okay. So you have to do the fragmentation of the blocking DNA. Okay. Now fragmentation of blocking DNA, and then you have to do the denaturation. So in this case also you are making the double stranded double stranded DNA to single stranded DNA of blocking DNA. Whereas in the probe also you are doing the same thing. Double stranded probe is converted to single stranded probe. Now you have to add these two things, probe as well as you have to add this blocking DNA. Okay, so what will happen? Where you have to add? Already you have prepared the slide. On the slide you have added your sample of interest. That means if you are taking any genome, you want to sequence or you want to identify the genome of the plant cell, so you just add all few plant parts, leaf parts you just add, or any other part of the plant you can add here, or tissue from the animal you can add, the cells from the animal you can add here on the slide. Now what will happen? On the slide you are adding, so on the slide you have the genome where you want to identify, which genome you want to identify now or localize. Now what will happen? You have added probe which is labeled with the fluorescent compound, and simultaneously you have to add blocking DNA. Okay, so these two you have to add it. Okay, this is the sequence genome which is present, hybrid sequence if you are trying to identify which hybrid is there. So what will happen? Probe will bind with the complementary sequence here. This is the probe binding. Whereas if you consider another one, that is the blocking DNA, this is a binding will be there. So the blocking actually unlabeled DNA. So there is what will you can observe through the microscope. So binding will be there. So there will be competition between the probe and unlabeled DNA binding. So finally, you can find few genome sequence, specific genomic sequence, which is just complementary to the probe DNA. You can identify that sequence. Okay, so let's see what are the steps involved here: direct and indirect probe labeling, then fragmentation of the blocking DNA, slide preparation, probe and blocking DNA denaturation in a hybridization mixture. Next step is the addition of hybridization mixture with the probe and the blocking DNA. So X are together you have to add blocking DNA, denatured blocking DNA plus labeled probe, denatured labeled probe. Now denaturation of the chromosome DNA, or you can do the denaturation together. After addition of these things, you can do the denaturation together. Now in situ hybridization of the probe and blocking DNA in the target sequence of the chromosome. Now detection of the probe in the chromosome DNA of one parent and in an indirect labeling. Now chromosome DNA molecule of the second parent associated to the unlabeled blocking DNA. So we are taking, if you consider you are checking this is one parent DNA genome, and this is another parent. If you are checking here, this is the hybrid offspring. If you consider, we are checking these two sequence. So in order to do that, chromosome DNA molecule of the second parent. The first one is detection of the probe in the chromosome DNA of one parent. So you are using probe against, if you consider this is the probe against one parent. Okay, this is one parent against which you are using probe labeled with the fluorescent. Whereas if you consider this is the green parent, another parent genome. So you are preparing probe. Okay, here also we are preparing short chain sequence in order to bind so that it is also making a hybrid. And in that case, labeling is not done. Probe type of sequence is there, DNA, RNA is there, DNA is there, single stranded, but labeling is not done. So one parent you can consider ki label is there, another parent not done. Okay, so you are using the reference one, that is the labeling. Now, chromosome DNA molecule of the second parent associated to the unlabeled blocking DNA. So, in this case, okay, if you consider the green one is unlabeled one. Now, visualization of hybridization signals. So, we are visualizing this part. Okay, visualization of hybridization signals associated to a probe. Okay, in this case, green color. In this picture, actually, green color fluorescent compound is utilized. Okay, now unmarked chromosomes are visualized with a counter staining. So if you if you want to visualize second one, which is not labeled, you can do the counter staining. So if you do the counter staining, what will happen? Blue color will be for the second parent, which is actually blocking DNA. Okay. So in this figure also same thing is given and same steps are given, nothing else. So when the probe labeling is direct, the detection step of the gas can be excluded and the fluorescence are the signaling molecules and can be directly visualized in a fluorescent microscope with the appropriate filter. Okay. So finally you have to visualize now, that is through the microscope, fluorescent microscope. Already I have discussed about the fluorescent microscope. So in this case, a background will be black and you can visualize in, if green color is given, you can visualize green colored fluorescent compound. Okay. So here also same how in situ hybridization is done, and this is taken from another source. See here, probe DNA labeling. Either you do direct labeling or indirect labeling, and you just do the denaturation. Okay, you have to do the denaturation, then only you can get a single strand. Now another thing you have to take, that is the blocking DNA fragment I told you. So in that case also you are doing the denaturation. Okay, blocking DNA fragment. No fluorescent compound is attached with this probe, a blocking DNA fragment. Now similar way you have to do the denaturation. You have the single strand. Now you have to add all probe plus blocking DNA fragment in the slide, or after addition of the double stranded probe DNA and blocking DNA, you can do the denaturation in on the surface of slide, because on the slide genome also you have to do the you have to make the single strand. Okay. Now if you consider this is the chromosomal DNA you want to identify, what will happen? You have to do the denaturation of the chromosomal DNA. Okay. And what will happen now? In situ hybridization is happening, because in this case probe DNA is there and blocked DNA is there, blocking DNA is there. So two types of combination you will find, two types of hybrid you will find. One will be labeled with the fluorescent compound, another will be labeled with without labeling. So this is actually labeled one and this is actually not labeled. So those are, this is actually blocking, that is not labeled with anything. So blocking DNA attachment is that you can visualize that thing after the counter stain, after that. Okay, but in this case you just visualizing the probe one, which is attached with fluorescent compound. You are visualizing this part only, probe detection. So probe DNA labeling molecule, this is the labeling molecule, you just visualizing that. Okay. So these are the steps involved in GIST method, genome in situ hybridization. So genomic in situ hybridization is a cytogenetic technique that allows one to radio label parts of genome, or you can do the labeling with the radioactive compound. In this case we have discussed this, or the labeling is done with the fluorescent compound. You can simultaneously do, not simultaneously, if you don't have a fluorescent compound, if you have radioactive compound, you can label with the radioactive. So last that probe preparation will be different. If you are using radio label, you have to follow separate pathway to label the probe, otherwise everything will be same. Okay. Now, GIST was mainly developed for the animal hybrid cell lines. So, whenever in laboratory you are making hybrid cell line, in order to identify that whether hybridization is done or not, proper hybridization is not done or not, you can analyze that. Later used for the plants at Plant Breeding Institute. So, initially it was started with the animal hybrid cell preparation cell line, and next it is actually popularized in Plant Breeding Institute, Plant Breeding System. Now, GISH is quick, sensitive, accurate, informative, and comparative approach rather than absolute one. Absolute approach for the cytogenetic technique. As compared to normal cytogenetic technique, this is very quick, sensitive, accurate, informative. Okay. Now, GISH technique is an advancement in the fluorescence in situ. This is, I told you, fish.
Our students in native, so there's no class today.
Okay, the technique involves, so what is the principle here? We have discussed already the same thing is a revision. See, the technique involves the extraction and subsequent radio labeling or fluorescent labeling of whole DNA of one organism and to use as a probe to target the genome of another organism. Okay, so you can compare key species 1 and species 2. There is sequence similarity is there or not. Now, parts of genome that are sufficiently similar to the probe hybridized to form a probe target complex, which is now labeled. Now, the remaining unhybridized parts of genome can be further tend to view them. Okay, so this is the technique. Again, I have written, I don't know why. And this is main steps. These are the main steps. Separately, it is written direct and indirect labeling, blocking DNA fragmentation, preparation of slide, denaturation of probe, everything same. It is mentioned here. Whatever I have discussed, already discussed these all the steps. Okay. Now you can visualize this counter stained one. This is labeled probe. You are labeling it with the green one. Now merge, you just merge two image. You will find key which one is stained, which chromosome is labeled, or which chromosome is not labeled. Okay, so you can compare that. Just merge the few chromosome cannot be labeled to using the same probe. Okay. Now let's discuss the what is the difference between gish and fish. Okay. Now genomic in situ hybridization, that is actually in this case probe is entire genome. Okay. Whereas comparative approach is done here in the genome, whereas if you consider the fish, in this case probe is oligonucleotide sequence. Now comparative approach is taken care of in case of gish, but in fish you have to do the absolute approach. A comparative means we are doing the comparison. Na? Here counter staining is done by the labeled DNA and we are comparing it. Comparative study is happening only in case of gish. But in fish you are doing directly identifying the chromosome, any sequence inside the chromosome. Okay. Now, main application of GISH is establishing polygenetic relationship, evolutionary study, and main application for the fish, that is determination of repetitive DNA sequence. If there is any repetitive sequence, I told you telomere, centromere. Since it's ma'am wants request, can we please continue this tomorrow, ma'am? I need to. Okay, it's done, already done. It's over, beta. It's over. You can leave because 7:30 is already done. Already over 7:30. So nothing else. Already everything I discussed separately written, separate sentences is given. Nothing else. Okay. So you can leave. Different applications are given here. Otherwise, shortly I have discussed what are the applications of GISH and nothing else. I will share this PPT. If you want to check, you can check it. Okay. So your class is over. You can go, Bhavna. Enjoy your Navratri. Okay. So I hope you understood the difference between gish and fish. Okay. Don't be confused with these two things. Okay. 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 Chalo. Now your health is well, na? Okay. Chalo. So this all about fish and dish. Okay. You just enjoy the Navratri. Okay. Tata. Good night. Bye. Okay.